epistasis or know what epistasis is. It would be a application problem solving scenario. Now, I am going to caveat what I've said. I am absolutely convinced about the whole OCR A and the AQA situation with these topics. Any other exam board, I am going to say to you, double check with your teachers and check out exam questions. Your teachers should be able to help you. But let's get going. So, a few characteristics that you might remember back from your GCSE are controlled by a single gene with just dominant and recessive alleles. And this whole, the rules of genetics were outlined by Gregor Mendel before DNA and chromosomes were even discovered. I mean, the most extraordinary contribution to biology by Gregor Mendel. And you might remember some of these single gene characteristics, you know, the good old straight thumb, curly thumb, or the shape of your thumb, whether your earlobe is attached or unattached, whether you have dimples when you smile, whether you can roll your tongue, all of those kinds of things. But at A level, we also learn about how different variants of characteristics are inherited when two genes are either on the same chromosome or on the same, what I'm going to call normal chromosome, we'll go into it in a minute, so that is autosomal linkage, or on separate chromosomes, dihybrid inheritance. And we're going to go on to talk about when two genes interact, where the alleles of one mask the effect of the other gene. This is epistasis. So let's do a quick recap, and it is going to be a quick one. I am going to assume that you remember what genotype and phenotype is. Allele combinations, what the organism looks like. Dominant and recessive, only one required, two required. Heterozygous and homozygous, mixed up, the same. But I do want to go through again what a gene and what an allele are. Gene, section of DNA that codes for a protein that determines a particular characteristic. Thumb shape, fur or hair color, whether the wings of a fruit fly are normal shaped or slightly differently shaped. Those kinds of things. Whereas an allele is a different form of a gene, and that comes about as a result of mutations and only mutations. And these cause variations in those characteristics. So thumb shape, whether your thumbs are straight or silly curly, whether your fur or whether an animal's fur is brown or black, whether hair is blonde or brown. And for flies, whether their wings are normal shaped or vestigial shaped. Vestigial actually means so small that they're practically useless. And I just want to remind you what homologous chromosomes are, because this is something that is often confused. This is when the paternal and the maternal chromosome of the same type pair up together. We're talking about the chromosomes that have the same genes on them, and those genes will be in the same loci on the maternal and the paternal chromosome. So here are some silly pictures of chromosomes where the DNA has already replicated, and here we have our two homologous pairs. Each homologous pair being made up of one paternal and one maternal chromosome. Here is a single chromosome, but it has two chromatids, two sister chromatids, and it's joined at the centromere because the DNA has already been replicated. If you are ever in doubt looking at a load of diagrams of X-shaped chromosomes and you need to know how many chromosomes there are in the picture, count the centromeres. There's a nice simple rule for you. So if I said that these were the alleles of gene A and gene B on the paternal and the maternal chromosomes, can you easily work out what the allele would be here and here, here and here? Hopefully, you're recognizing that because the DNA has just replicated, the alleles on both chromatids will be exactly the same. Okay, quick recap on meiosis. Meiosis, my GCSE definition, 1 DNA replication and 2 cell divisions. Those cell divisions include both the nuclear division and the cytoplasmic division. So the nucleus, that is when the genetic material is split between the cells, and it is split twice so that we go from an original diploid cell with pairs of chromosomes to 4 gametes, each one with only 1 chromosome from each pair in the gamete. And these 2 cell divisions that make up meiosis are called, funnily enough, meiosis 1 and meiosis 2. Now, in meiosis 1, the homologous pairs of chromosomes are separated. The homologous pairs line up at the equator and they are separated to the poles. In meiosis 2, the chromatids of each duplicated chromosome are separated. And as soon as those chromatids separate, it's just like a child leaving home, they suddenly become an adult. So when the chromatids separate, you can call them chromosomes because they are, in effect, this is going to form the 2 chromosomes, 1 chromosome from each pair going into 1 gamete. You can talk about chromatids throughout your A-level answers. Don't worry about that whole change from chromatids to chromosomes. But also in meiosis 1, 2 other really important processes happen. And that is crossing over, and that is when the, what have I done here? So yeah, that is when portions of non-sister chromatids exchange portions of those chromatids. So maybe I should just say that simply, when non-sister chromatids exchange portions. I'm encouraged, thank you. Sorry, who is that? Scope source, thank you. So if we have these alleles, which we'd already talked about in the previous slide, can you see that actually you're going to get a mix-up in the alleles? On this chromatid that is going to end up being the 1 primarily paternal chromosome in a gamete, you're going to have a little bit of the maternal chromosome on it. And therefore, whatever alleles are on this end of the chromatid, it's going to be mixed with some of the maternal alleles. It gives rise to, it is an exchange of alleles, an exchange of portions of the chromatid between 2 non-sister chromatids, and it gives rise to a huge variation of alleles in the gametes. And then we have the process of independent assortment. And that is most clearly seen during meiosis 1 when those homologous chromosomes line up at the equator. Because whichever way round 1 pair of chromosomes line up at the equator, whichever way they assort, it is completely independent to any of the other chromosomes that are lining up at the equator. It's why it's called independent assortment. You could have the paternal and maternal chromosomes in that order, or it could be the other way around. for each and every pair of chromosomes. So hopefully you can see that if there was only one pair of chromosomes in a cell, there would be two possible combinations in which the chromosomes could be split between the two first cells that are produced as a result of meiosis 1. Scope source, I will talk about that in a minute. Let me just stay on this for a moment. But does it make sense that if you had two pairs of chromosomes in a cell, actually the number of possible combinations are 4? That's 2 to the power 2. The power 2 being the number of chromosomes that you've got in the cell. So this increases the combination of chromosomes and alleles in the gametes. So can you see that for the human chromosomes, for human cells, we have got 23 pairs of alleles. There are 8.4 million possible combinations in which our pairs of chromosomes can be split when meiosis happens in either the ovaries or the testes. Huge variety. This crossing over particularly applies to autosomal linkage. And independent assortment, it does apply to autosomal linkage, but I'm really going to emphasize its application to the variety of gametes that are formed when we talk about dihybrid inheritance. So there's just a heads up at the start. So let's talk about dihybrid inheritance. Really importantly, this is when two different genes are on separate chromosomes, different chromosomes. So here we have an example here, and this cell is heterozygous for both genes that we are looking at. So we're actually going to use the example of body color and wing shape in the good old fruit fly, probably one of the primary organisms that is used to investigate inheritance and genetics, Drosophila melanogaster. And this is a little picture. It's those little fruit flies that buzz above the fruit when it's a little bit overripe. That's them. So here are the alleles. We've just got dominant and recessive alleles, no shenanigans with multiple alleles or codominance or anything. So, and this is G is the color of the body and N is the shape of the wings. So here are my headings for working out the inheritance problem. And I'm not going to apologize for using these. This one is the most important one that you've got to get right. If you can get the possible gametes correct in your inheritance problems, forming the Punnett square and working out what happens in the offspring falls into place. This is where students go wrong. So watch out for this, particularly when I talk about the F1 cross. We'll go into it. So I am going to be crossing purebred grey normal, so grey-bodied normal wing Drosophila with black-bodied vestigial wings for Drosophila. So those of you who are not in live stream land, stop the video and see if you can work out what happens here. And then I'm now going to go through it. So I've said that they were purebred. That means that they are homozygous for each gene. So we have got this genotype and this genotype. Now remember, the genes are on different chromosomes. So strictly, I should be able, and because of independent assortment, there are potentially, let's just, say that these are, that I'm just going to sort out this one first. So just imagine that they're both big G and they're both big N. If I were trying to systematically sort out the allele, sorry, if I was systematically trying to sort out what gametes could be formed, I would systematically go, right, it's big G and big G and big N. Big G and big N. Oh, let's do that one again. Big G, big N, big G, big N. Funnily enough, all big G and big N. Notice that every gamete has to have one allele from each gene. That will stop you writing this as a gamete. Clearly that can't be the case. You've got to have at least, you've got to have one allele from each gene in your gametes. So here are my fine diagrams. I'm just pretending we're now pretending that this is the male, this is the female, really doesn't matter. That we have gametes. All of the gametes from this individual will be big G, big N. There are no other options. And hopefully you can follow that all of the gametes here will be little g, little n. And therefore, hopefully, this rather simple Punnett square will show you that all of the offspring are going to be heterozygous for both genes. But phenotypically, they're all going to be grey-bodied and normal wings. Now, one thing I want to just point out, as a convention, when you put together when you're putting together the possible offsprings in a Punnett square, when you're using dihybrid, you're dealing with problems with two genes, the convention is that you always put the dominant allele first, and you always pair your alleles. It will make it far easier for you to identify the different phenotypes if you do that. So all of the F1 means first filial generation. Filial is talking about brother, sister. So it's talking about that first generation that is created. And phenotypically, they are all grey-bodied and all normal-winged, even though they're heterozygous for both. So now we're going to go on to do an F1 cross. And now we need to really systematically work out the gametes here. Because of independent assortment, we could have all of the possible combinations. So for both of these, here are all the possible combinations. And it's as a direct result of that independent assortment. This pair of chromosomes either being this or this on the equator during meiosis 1, and this little pair of chromosomes either being this or this as well, but it being completely independent. And as a result, now we have that hugely complicated Punnett square, which I desperately tried to draw out neatly. Notice that when I am doing inheritance problems with two genes, I put little dashed lines in my Punnett square to make sure that I am making it really clear to my brain that each one of these is a gamete, and I don't suddenly start thinking about it as a genotype of a monohybrid cross or something. I don't just get confused. So that tells me that I've got to put both of these together. It's just a silly little thing that I do to make my brain work clearly. And therefore, if we sort out all of the different possible genotypes from this Punnett square, we can categorize them. We can categorize them where there is at least one dominant allele for each of the genes, because of course, if there is, these dashes here mean that this could either be this or this, because actually it makes no flipping difference, because if you've got one dominant allele, you know what the phenotype is going to be. That's what these little dashed lines means. It means we've got one dominant allele, the other one makes no difference to the phenotype, So hopefully these different genotypes make sense. these different phenotypes make sense, and if we add them all up carefully, we will end up with that classic 9:3 to 3:1 ratio. So remembering, and here you need to remember that everything that you draw out in your Punnett square are the possible outcomes from a single fertilization. So if there was just one fruit fly made from a mating, of course there wouldn't be, they lay hundreds of eggs, you know, but if there was just one fruit fly, you could say there is a 9/16 chance of it being grey-bodied and normal-winged, a 3/16 chance of it being grey-bodied and vestigial, 3/16 chance of it being black and normal-winged, and a 1/16 chance of it being black-bodied and vestigial-winged. If, on the other hand, you do, as they do with fruit flies, and bring about a cross where hundreds of offspring are made, therefore, we can then count up the offspring and see if they follow this kind of proportion. And the larger the number of the offspring, the more close to this ratio they will become. Yes, sorry, let's scope. I will, scope source, I will answer that. Shortening of the chromatids, yes, because each chromatid, this is off topic, each chromatid has a telomere at each end. So that's a little bit of extra DNA. It's actually a repeated sequence. And each time that the DNA replicates, a little bit of that telomere is lost. And therefore, every time the DNA replicates, the DNA shortens very slightly. And when the cell runs out of telomere at each end of the chromosome, the cell dies. And that is thought to be one of the contributors to aging. So hope that helps. Okay, back to dihybrid inheritance. I think we have finished dihybrid inheritance, and now we're going to go on to autosomal linkage, and I'm going to use exactly the same example, but I'm now going to pretend, oh sorry, what I should have said, this is autosomal linkage. An autosome is a normal chromosome. In other words, any kind of chromosome except for the sex chromosomes. So in humans, we have 22 autosome pairs and 1 sex pair of chromosomes. The first 22 pairs in our karyotype, don't worry about that word, are autosomes. The 23rd pair of chromosomes are the sex chromosomes. So here we have genes on the same autosome or somatic chromosome. So I'm going to use exactly the same example, but now I'm going to pretend that instead of these genes, these 2 genes being on different chromosomes, they are linked, they are stuck together on the same chromosome. We've just got that single pair of chromosomes, the long chromosomes, paternal and maternal chromosomes. And now let's go through the same problem using the same headings. And again, if you're not in live stream land, stop the video and actually have a go at doing this yourself. So back with the purebred parents. But now notice that when I am working out what the possible gametes are, I draw a stick, because I want to be sure that I'm constantly keeping in mind that these genes are stuck together. So therefore, there is only one type of allele for each of the parents. And therefore, once again, we're going to end up with, in the F1 offspring, of all grey-bodied normal winged flies, but all heterozygous for the 2 genes. Now it gets more interesting, because now what happens if we do an F1 cross? Well, this is what happens. Look at the arrangement. So we've got big G, big N on one chromosome, little g, little n on the other. So this is assuming no crossing over. I will talk about crossing over in a minute. But now, can you see that these genes, sorry, let me reword that. Can you now see that these alleles are stuck together? They ain't moving apart from each other. We're not shifting them around and making lots of different combinations. There are only two possible gametes for this F1 cross. And therefore, we end up with a much simpler Punnett square than we did with dihybrid inheritance. And notice what the phenotypic ratio is. The phenotypic ratio for the dominant alleles, phenotype, grey-bodied normal winged to black-bodied vestigial wings, is 3:1. Now that might ring a bell, because that's the ratio that you get if you cross monohybrid heterozygotes. And does it make sense that because these are stuck together on the same chromosome, that is what would happen? I'm hoping that you're seeing the link there. Shri, how do you know which, oh, brilliant, well done, Shri. How do you know which alleles are on the same chromosome when they give you the outcome of a phenotypic ratio? So what you're saying is that you don't know whether this might be big G, little n, and little g, big N. Right, in an exam paper, they will draw you a diagram, okay? If they make it absolutely clear that it is autosomally linked, they say that the genes are on the same chromosome, and for some bizarre reason, they don't show you a diagram, assume that the dominant ones are on one and the recessives are on the other. I have not seen a situation where they don't actually draw out the chromosomes so that you know which alleles are stuck together. It's a really valid question, Tree, and I'm delighted that you asked it. And I'm hoping that I've answered it, hoping that I've answered the question that you think you're answering. Right, okay, back to the plot. Right, so far I have assumed no crossing over, because of course this could get split, this could get cut and pasted, so to speak, during crossing over. Now, if crossing over occurs, instead of, let's assume that we did, you know, we got about 1,200-ish offspring of dear old Drosophila melanogaster, and instead of getting this classic and recognizable 3:1 ratio that we would have expected from autosomal linkage, these are the kind of numbers that you would get. Notice that the dominant phenotype, what I'm going to call the double dominant phenotype, and the homozygous, the double homozygous recessive phenotypes still have a recognizable ratio of 3:1. But you've got these, what I call the mixy-uppy phenotypes. That's really technical speak, isn't it? We've got a very small number of mixy-uppy phenotypes to kind of screw up our 3:1 ratio. This tells you that crossing over has occurred, because in a few cases, when the gametes have been formed, the crossing over has occurred between the loci of the G and the N genes. And can I also point out that if the distance between the two genes is very small, the chance of that cutting that happens during crossing over, the chance of that happening between those two closely placed genes is very small. you're going to get a very small number of mixy-uppy phenotypes. If, however, the distance between the two genes is very big, there's a far greater chance that crossing over will occur between those two genes. The larger the number of mixy-uppy phenotypes, the greater the distance between the two genes on the chromosome or chromatid, if that's what you're wanting to be thinking about. I hope that makes sense. Is there anything else that I want to say? Yes. Oh, look, I was just saying here, look, closely linked. This is indicating, the very small number is indicating that these two genes are actually closely linked on the chromosome. Okay. All right. I just want to really hammer home the difference between dihybrid and autosomal linkage. So remember those F1 crosses that we've just gone through. Hopefully you remember that for dihybrid, when we crossed those double heterozygous parents, heterozygous in every way, we got, for dihybrid inheritance, when the genes were on different chromosomes, we got the 9:3 to 3:1 ratio. But when they're on the same chromosome, we get the 3:1 ratio. Remember that this is what I call that double dominant, when both genes have a dominant allele. And this is where, when both genes are homozygous recessive. And again, and this is absolutely as a result of independent assortment. The autosomal, notice that this is when, we get those two dominant alleles for each of the genes, and this is when we get the homozygous recessive for both of the genes. This is when there is no crossing over. But notice that we get this ratio equally because of independent assortment. But the independent assortment has stuck, my fingers being the chromosome, has stuck the two genes together. The genes have moved with the chromosome. They haven't been split. They haven't got any other allelic variation unless crossing over occurs. This is obviously the ratio when no crossing over occurs. So just really quickly, and I'm going to fly through this, what about if this cross occurred? This is called a back cross. So you've got an organism with the dominant phenotype being expressed. It's grey-bodied, it's normal winged. Who knows whether it's, heterozygous or not, possibly. If you do a back cross, you'll find out. And a back cross is when you cross it with the homozygous recessive individual. So what would happen if we did this under dihybrid rules and we did it under autosomal rules? I'm just going to fly through this. Possible gametes, hopefully we're getting that. Hopefully we're understanding loads of possible gametes under dihybrid, but only two possible gametes for the heterozygous one under autosomal. And therefore, this is the ratio of the offspring that you get. Notice that you get every phenotypic option under dihybrid, and you don't under autosomal. Under autosomal, you actually get exactly the phenotypes and the genotypes of the parents. Here, I should have put equal proportions, and I should have put of all phenotypes. You get every phenotype and you get equal proportions of them. And the rule here is that you always get a high proportion of the offspring having the parent's genotype and phenotype, and therefore phenotype, even with crossing over. Because remember, with crossing over, you only get those very small numbers of mixy-uppy phenotypes. So the ratios are different because you get all of the phenotypes here in equal ratios, whereas here, even if you do get all of the phenotypes, It's fundamentally one-to-one with a few silly little individuals of the mixy-uppy phenotypes in there as well. Okay, really quick. Do have a look at my website, because I've got all of my videos on there, and every video has exam questions at the end, which I obviously don't do when I'm on live stream. All of the videos or the whole course for 50 quid, I'm going to get everything completed by Christmas. So if you subscribe, you're going to get everything for when you need it. You could even pay for your own subscription, because you can earn 10% commission on any friend's purchases. And there's a freebie. Do have a look. And I'm just going to quickly show you that I am adding new videos to the list. This is the year 2 list. I'm constantly uploading the new recently made videos. And actually, I know there's still a lot of gaps, but I'm going to get through those gaps really quite quickly. So have a look at my website. And finally, woohoo, epistasis. 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 Oh, little bit of roots of words. Epi means on top. Epidermis, the layer of cells on top of a whole load of cells on top of your skin. Yes. Epi means on top. Stasis means standing. So this literally means standing on top of. So this is when one allele combination of one gene, because we're still talking about the inheritance of 2 genes, the effect of 2 genes, when the allele combination of one gene masks the effect of the second gene. And can I just say, for the purposes of A-level, you always assume that these genes are on different chromosomes. Okay, so we're going to do the rather nice example of how coat colour in Labrador dogs is inherited, because they're cute. So there are 2 genes that decide which coat colour. One gene decides whether pigment is made or not, whether there is any kind of pigment. And that we're going to use the notation of E for this gene. The dominant allele causes pigment to be made. The recessive allele means that there is no pigment. Homozygous recessive, no pigment, you get a golden Labrador. Yeah, sure it would be. Well, I still reckon, I reckon you could still do it. Just apply the principles, but I'm not even going there, Okay, the second gene decides if pigment has been made, whether that pigment is black or brown, giving rise to either a lovely black lab or a brown lab, commonly called a chocolate Labrador. So here we go. Dominant allele is black, recessive is brown. So let us do a cross. Those of you not in live stream land, have a go at doing this. Work out what the phenotype is. What's the dog going to look like? What are the possible gametes? We're dealing dihybrid inheritance, remember. What are the possibilities for the offspring? Okay, let's have a go. Well, I'm looking at this and I'm going, both of these, oh, that was skipped a bit. Both of these are going to be black labs, but they're heterozygous for both genes. And therefore, we get the full combination of alleles. Big B, big E, big B, little e, little b, big B, little e. Can't do it. Little b, big E, little b, little e. Okay, so we get the horrendous giant Punnett square, which I'm not drawing out again. But notice we get the same possible phenotypes. Notice that, again, remember that these dashes means that this could be either big B or little b. It makes no bogging difference. This is, so the big E means that there's pigment. The big B means that it's going to be a black lab, yeah? So here we go. Black lab. Notice that wherever we've got the homozygous recessive E gene, it masks this B gene that decides the color, because it's basically saying, hey, we ain't making pigment, so it's just going to be golden. And notice now what the ratio is. It is a 9:3:4 ratio. Notice how this is being derived from the 9:3 to 3:1 ratio. Notice the link. From black to brown to golden lab. Okay, notice how that links in. Now, this is called, because the E gene is the epistatic gene. It's the one that is standing on top of the second gene. It's the one masking the second gene. This is called a recessive epistatic gene because it's only when it is homozygous recessive combination of alleles that causes the masking of the other gene. This example here, and this is actually what happens, it's a recessive epistatic gene. Now, can you work out what would happen if, and those of you not in live stream land will be more capable of doing this because you can stop the video, if we pretended that it was the dominant allele that caused no pigment, and therefore it was a dominant epistatic gene? Can you work out what the ratio would be now? Have a look at this outcome and you should be able to work it out, but even those of you in live stream land. Does it make sense that the ratio now is 12:3:1 because any genotype with a dominant E allele is now the ones that are golden? So we have got 9 plus 3 equals 12, and then we would have 3 that were black and 3 that were brown. So, and again, notice the relationship between this ratio and the 9:3 to 3:1 ratio. AQA students, you have to know what epistasis is. You do not have to know the difference between dominant and recessive epistasis. You don't have to know those terms. But you, if told that, you know, if you're basically told how a couple of genes work in terms of their epistatic effect, you have to be able to work out what the ratio is, but you don't have to use dominant and recessive epistatic terms. OCR A, you are expected to know that. I hope that helps. Oh yes, and absolutely, Shri, exactly like enzyme pathway, exactly. If there was an enzyme pathway where A goes to B goes to C, if the gene for the enzyme here doesn't work that turns from A to B, you're never going to get C, yeah, because this gene for the enzyme is epistatic to this one, yeah, because it masks, doesn't matter whether that enzyme's there or not, this one is going to mask the effect. Hope that makes sense. Sorry, that was a completely off the cuff explanation. Okay, here is my inheritance map. It's not perfect, but I'm really hoping that it might just help you clarify all of the different types of inheritance that you have had to learn about. So I've separated it for convenience into whether we're dealing with one gene or two. Monohybrid is just when you're looking at when you've only got one dominant and one recessive allele, so therefore you can use big A and little a because of course you've got enough symbols to denote dominant and recessive. And here is me denoting the heterozygous cross, which gives rise to a 3:1 ratio. Notice that multiple alleles, I've underlined multiple because it means there are lots of different alleles, but notice that only 2 alleles can be carried by any individuals because our chromosomes come in pairs. Oh, Christy, what is the similarity difference between monohybrid and dihybrid? Monohybrid, you are only looking at one gene. Dihybrid, you are looking at the inheritance of 2 genes at the same time, and if we are specifying dihybrid, you are telling me that they are on separate chromosomes. Autosomal, you are looking at the inheritance of two genes for different characteristics, but they are on the same chromosome, same autosome. And epistasis is an offshoot of dihybrid because one gene, or the allele combination of one gene, if I'm being precise, masks the other. I'm also just wanting to point out why you have to use this really irritating notation of using all these superscripts, these little letters at the top. Well, that's because they are multiple alleles. We've only got big A and little a. You can't denote more. There are just multiple alleles. And the same here, which is why superscript is used, because there isn't a dominant and recessive. Both alleles contribute to the phenotype. Sex-linked, you have to denote your X and your Y chromosome, because you've got to know which allele is on which chromosome. But notice sex-linked means that the gene is on the X chromosome only. You assume that there is no allele on the Y chromosome. And here I just wanted to point out the inheritance of sex-linked conditions. It is passed from the mother if she's a carrier or a sufferer. If she's a carrier, she will only pass it to her son. In fact, if she's a carrier or a sufferer, she will only pass it to her son, and her son will pass it on to his daughter. So it zigzags through the generations from mother to son to granddaughter to great-grandson. Hopefully that's helpful. Okay, what is a hypostatic gene? Hypo means under, static. The hypostatic, if you're talking, the hypostatic gene, I've never come across that term, I'm going to be honest, but logically, the hypostatic gene is the one that is being masked, because hypo means under. So just me knowing roots of words helps me work that out. If you're using that term in relation to epistasis, the epistatic gene is the one that does the masking, and the hypostatic gene is the one that is masked. I've never seen that in an exam answer, so don't get hung up about using that term. Oh, there you go. Yeah, thank you, Shri. Please, that was confirmed. Okay, I think flipping heck with there. This has been a real marathon.